before Jack, before David, you were from 1986, correct?
Yeah, 87, yeah.
87. So Dr. Bruce Jenkins is a physical chemist and an expert in spectroscopy, MR spectroscopy, but he knows all the different modalities of MRI, magnetic resonance imaging. And one of the first people that with Jack Beliveau was doing fMRI experiments. So we have Risto here and his team, Bruce. And I think you tell us also some jokes about Jack so that we will have this condensed experience with some emotional memories that we can share with you also. So
Okay.
feel free to say whatever you want. Thank you
All
so
right.
Um,
Yeah, I noticed that I don't have a link to, okay, present. All right, so I've got to present. So let me get my slides up. Hold on a second, I'm sorry. Whoa, okay. I'm so sorry here. For some reason, the doc is not open, but here it is. Okay, so okay, so
we can see your presentation very clearly on you.
okay, all right. So so today I'm going to give you a wait a minute. Do I have the right slide here? Yes, I do. But
it was okay. Where did you click?
I got the wrong title. Anyway, this was my title from last year. I apologize. But this year, because there's less time for me to present, I'm not going to be presenting anything about magnetic resonance spectroscopy. But I will be talking a little bit about kind of general principles of T1, T2 for MR imaging, structural imaging, and then I'll cover a lot about functional imaging in the brain. So basically, there's kind of, I break it up into two kind of branches of MRI techniques. We have metabolic imaging, which can include magnetic resonance spectroscopy, which allows us to probe any number of molecules in the brain, changes in basal metabolism. And then there's the hemodynamic aspect of the metabolic imaging, which is cerebral blood flow and blood volume, and BOLD, which is blood oxygenation level dependent contrast, which is the kind of the standard tool used for fMRI, although some studies do use measurements of cerebral blood flow. And then we have the anatomic side, which is imaging of MR contrast using different parameters and knobs that we can tweak. So there's T1 and T2, which are intrinsic magnetic resonance relaxation times, proton density, magnetization transfer contrast. Then there's also diffusion, and that's making the images sensitive to the motion of water molecules. And there's a number of different ways to perform this. And lately, there's been a lot of very beautiful studies where they look at fiber tract mapping in the brain. And there's the idea that we're going to try and develop the human structural connectome. So our lab has just installed a second generation connectome scanner, which has incredibly high gradient duty cycles, as well as B values that we can attain, very large magnetic field gradient values we can attain to get ultra high structural resolution for looking at fiber tract mapping. And I'm sure you'll hear about that elsewhere in the conference, so I'm not going to cover that in great detail. And then we can look at what we call connectivity, and there's functional and structural connectivity. And so there's hopes that we can actually one day be able to unite the function of the brain using MRI and other techniques, electrophysiologic techniques, magnetic encephalography techniques, combined with the structural connectome to be able to start getting a much better map of brain activity. So of course, MRI, as we know, is more than just anatomy. I'm kind of reiterating what I just said, but you can get the anatomy. And what's great about MR in contrast to CT, in CT, you really only have one parameter, tissue parameter to deal with, which is density. In MR, there's any number of properties of water that allow you to generate contrast. And like I said before, you can do chemistry and you can do function and see changes in pathology. So in this CT, you kind of have, this is where you're at with CT. You've got this, cool looking car. You can look at density and dose of X-rays really as the only two parameters you have much control over. In MRI, you have a much more complicated cockpit to drive the scanner. And you can look at molecular composition, gradient trajectory, magnetic properties of the tissue, for instance, caused by paramagnetic molecules like deoxyhemoglobin. You can look at chemical exchange, which is the exchange of OH protons, for instance, off and on water. You can also change things like the scanner timing of the data collection. And of course, T1 and T2, which are kind of two of the main contrast knobs that we use in MR. So just to go over this, and I apologize if this is too basic a level for some of you, but this is just to kind of understand how we generate the contrast using these different parameters. So T1 imaging is usually, it's going to be contrast generated based on the T1 of the tissue. And things that have short T1s are things like fat, or things that have that are near a paramagnetic molecule like manganese or iron. And then you can change the what's called the TR parameter and generate more or less contrast based on T2. So you can see here in the curves that you have the curve for fat. And when you're changing this TR value in the scanner, you're making it sensitive to T1. And CSF has essentially the longest T1 of anything in your body except for bone. And fat has...
We cannot see any slide of yours.
You cannot see the slide?
No. We're seeing you.
Wait a minute. You mean I've been talking this whole time with no slides?
Yeah, because you had it. I thought it was a joke of yours.
No. Okay, I'll try it again. Let's see. I thought I was presenting.
Good joke, Bruce.
Well, I thought I saw a soccer ball coming at me, and then it hit me. Okay. So can you see that?
Yeah. Now we can see the how do you call that? Living room. Yeah.
Okay. Yeah. All right. So this was what I talked about in terms of the phylogenetic tree of MR techniques. I wish you had told me sooner. So I talked about this, but here's the slide showing you what I was talking about. Always, a picture is always worth 1,000 words. As you see, we can break things up to metabolic anatomic MRI. We have a lot of contrast knobs that we can deal with. We can look at structural and functional connectivity by uniting this BOLD or CBF with diffusion tensor imaging. So this is, again, just kind of reiterating what I just said. So this is kind of what I was talking about with CT. Can you see this now?
Yes. Okay.
So in CT, you kind of only have two knobs that you can deal with, density and dose. But for MR, you have a much more luxurious, cockpit to be able to tweak the contrast in your images. And you can look at things like the molecular composition of tissue using spectroscopy. You can change the gradient trajectories, which is how you manipulate a lot of the images that we do, and that's actually a source of a lot of different ways of altering the data and the contrast. Magnetic properties of tissue based upon, for instance, deoxyhemoglobin, as I was saying, which is paramagnetic. You can look at chemical exchange of hydroxyls, scanner timing and data collection. There's a lot of things that can be played around with there, and looking at T1 and T2 contrast. So this is going into, this is where I was before. So for instance, on the left you see is what happens to T1 tissues that have different T1s as a function of the TR. And what you can see is CSF, as I was saying, has the longest T1 of any tissue, except for a bone. But bone has very short T, you can't really see it easily with conventional MR. Fat has the shortest T1 of most of the major tissues that you see. And so what you can see is you can play around with the timing, scanner timing, which is this TR, the time of repetition, to generate contrast based upon the T1 properties of the tissue. So short TR gives you very strong T1 weighting, and a long TR gives you very low T1 weighting. For T2, we have kind of the opposite behavior, which is that fat has a short T2. So as a function of this other parameter, TE, which is the time to echo in an MRI pulse sequence, we can control the contrast due to fat and CSF. So CSF will persist in high intensity, even at very long TEs, and fat will decrease very quickly. So here's kind of a general rubric for what we see in the brain, which is that CSF has a long T1 and a long T2, so it's dark on T1 and bright on T2. Gray matter has intermediate T1 and T2, so it's darker than white matter on T1, but it's brighter on T2. White matter has a shortish T1 and T2, so it's brighter than gray matter on a T1, but it's darker on a T2. Fat has a very short T1 and short T2, so it's bright on T1 and dark on T2. Bone, as I said, has a very long, it's all but invisible except for the marrow, unless you go to highly specialized techniques, that are not likely to be used in a generic scanner. And then you have like tissues like the substantia nigra and the globus pallidus, and both of those regions of the brain accumulate iron and other paramagnetic metal ions for reasons that are unknown. But essentially, you can almost age someone by looking at the darkness in the globus pallidus, because the older you are, it just gets in there and it never leaves. So these are kind of generic 3 Tesla images. What you can see is there's the T2. So you can see the CSF and the spaces. You can see now, this is the globus pallidus. You see how dark that is, and that's because of all the iron that's there. As a matter of fact, if you took this same image of, say, a 15-year-old person, you would see that this globus pallidus would have the same contrast as the putamen. And it's only as you age that it gets darker and darker. And the darkening starts even by the time you're about 21 years old. This is a T1 image. And again, you can see that now because the white matter has shorter T1, it's got a brighter contrast than the gray matter does, which has a longer T1. And then this is just an example of these images looking at a stroke patient over time. This is taken in, and it's, so FLAIR is what they call a fluid attenuated inversion recovery. So this is basically a T2 image where you use the ultra long T1 of the CSF to null it using an inversion recovery sequence, it's called. And what you can see here on the T2 is you see a T2 bright here, which is where this stroke is. And you can see that it's evolving into a larger T2 bright area over 27 days. Here's what we call diffusion weighted image. And what this is this is giving you contrast based on the diffusion properties of the water. And in this case, I'll tell you this, what we know in a stroke is that the water that's inside the infarct area is what they call restricted. And it means the water cannot freely move around as much as it can. And that's due for a number of different reasons. That it cannot move around as much. So it doesn't sample as much of the gradient in the sequence. So it stays bright. So this is actually kind of the light bulb signature for a stroke. This is the apparent diffusion coefficient. So this is a scan that's sensitive to isotropic fluid motion. So you can see anything that has a high diffusion coefficient, i.e. that can move freely, which is like in CSF, can move almost the same as just distilled water. And you can see that it's very bright. And this is dark because where the stroke is, you have this restricted diffusion, so it can't move very much. And that's due to proteins that are there, as well as what they call cytotoxic edema, cell swelling. And so the cells get larger, the more water is inside the cells, and it can't move as much. And then this is the ADC image combined with a flare, where you get rid of the fluid, and now you can see this kind of matches better this area of cytotoxic edema. And this is kind of where you really have this the actual stroke. So you can see we have all these knobs that we can use to generate contrast in the images. We can also generate contrast using both diffusion and magnetization transfer contrast, which, as I told you, that's exchange of OH groups on and off macromolecules, and those things will alter the T1. And so if you have like a myelin sheath like this, what you can see is you can start manipulating the diffusion properties. This is called fractional anisotropy. And what this is indicating is that the water can move faster along the long axis of the axon than it can along the short axis. And so we can generate scans that are sensitive to either this, what they call fractional anisotropy, or radial diffusivity. And so, you know, you can use this in all kinds of situations. We just did, this is just some data we collected. I was going to show you just really to highlight the differences in these parameters. And we did this model of remyelination in a rat using a drug. But you can see that the T2, you can see this little area here in the corpus callosum. And here's a fractional anisotropy map. You can see, of course, that we get a lot going across corpus callosum. You can see decreases here where you've got this little lesion. This is a radial diffusivity, and you can see you can pick up this lesion in the magnetization transfer. Anyway, so there's just all these ways of generating contrast in tissue. And because this is supposed to be a general talk, I'm not going to go into any details about these studies. So a lot of my work involves either mapping receptors in the brain using MRI, or looking at neurodegenerative diseases. So we have all of these different control knobs, contrast knobs in MR that can be used to examine all these different tissue properties as a result of, for instance, neurodegeneration. So, but we're going to cover both the structural and the functional MRI, the overall basics of that. So let's start in with the function of the brain. And this is, you know, in 1812, this is kind of where we thought we were, 1810. They thought there were different parts of the brain that involved love of your offspring, a tendency to murder, cleverness. So they were kind of moving in the right direction in that, well, maybe the brain has specialized functionalities. It's just they were completely wrong about the functionalities. Poetic talent, I mean, it's just kind of a fantasy, you know, how you wonder how they come up with this stuff. And then this is kind of where we are more recently today, looking at things like the default mode network. So this is the part of your brain that's active when you're just kind of lying in the magnet, thinking about what you're going to do for dinner tonight, that kind of stuff. We have a somatomotor network, which is in B. You can see here, you can see visual networks, language networks, dorsal attention networks, frontoparietal control networks, ventral detection networks. And in addition, we can do all kinds of task-related activation. So we've come a long way, and we actually have pretty good handles on where most of the crucial functions in the brain are localized. And whoops. So the fMRI observables are BOLD, CBF, and CBV. So BOLD is blood oxidation. Excuse me? What? Is there a question? No. Okay.
Oh, it's something happened.
What happened?
Go ahead. Oh, I don't know.
You don't know what happened. Okay. What, you cannot see the slides or you can't hear?
We can.
Everything fine now.
Okay, good. Good. Okay, internet glitch maybe. So BOLD signal, which we'll cover what that is in a little bit, is non-invasive. It's got good volume coverage and good sensitivity. And this is by far the standard fMRI tool used. So probably, I would say, 95% of fMRI studies are using the BOLD technique. CBF is also non-invasive. but it's got lower sensitivity, lower contrast to noise than bold, and it's not, you can't really run as fast as you can with bold. So you don't get as good volume coverage per unit time. But it's also nice in that you know exactly what physiologic parameter you're measuring, which is blood flow, as opposed to bold, which is a complicated parameter, as I'll discuss later. You can also do functional imaging using cerebral blood volume, but this requires a contrast agent and actually has the best sensitivity compared to bold or CBF, and it's 2 to 4 times the contrast to noise as bold. But unfortunately, because it requires injecting a contrast agent, it's got limited use in humans. So the first study, you know, kind of to go over the origins of bold, it's based on the relaxation properties of water, which were, you know, kind of determined by Bloch, Solomon, and Bloombergen back in the 1940s and 50s. The first image of CBV by PET was in 1974 by Grubb. Arno Villringer in our lab was the first to really look at intravascular contrast agents and how they affect extravascular water. So this is kind of a very important finding, because what it shows, and this is important for both bold and for contrast agent use, which is that the magnetic field gradients that are induced in the blood vessels extend well beyond the blood vessels. And that's important because it means that you don't have a technique where you can just image the blood vessels. And as we know, the blood volume of the brain is only about 4%, so that would be a very small signal we're trying to image. But in actuality, because of the spread of the magnetic fields, we get a larger sensitivity than we would for just looking at an intravascular contrast agent that's only vascular. First gadolinium agent was approved for human use in 1988. This was primarily for looking at things like brain tumors and stroke and blood-brain barrier leakage. Not so much for looking at blood volume and function. That was really kind of pioneered in our laboratory. Bruce Rosen kind of was the first to apply the gadolinium technique to look at perfusion in the brain. And in 1991, Jack Beliveau reported the first ever fMRI studies using a contrast agent. And then Joe Mandeville and I, we pioneered this technique called the iron increase relaxivity for optimized neuroimaging, the iron technique, which can increase your contrast to noise for functional studies by a factor, like I said, of 2 to 4 over bold. This was Jack Beliveau. So I was asked to say a little bit about Jack Beliveau. So he was a very interesting character, as those of you who might have known him. Unfortunately, the year that he died, he was apparently nominated for the Nobel Prize for fMRI. So he actually invented the term fMRI and was instrumental in bringing together the team to do the first study that was published in Science in 1991. And that image is one of our collaborators in the lab, his brain, and that is the visual cortex that's been activated using sequential injections of gadolinium PDPA. And Jack was a force of nature in terms of his social abilities to organize a large experiment. He got so many people to work on this experiment because he got everybody excited by his vision. And what's interesting is his vision, which sounds it sounds very sentimental today and a little bit nutty, but his mother was elderly when she had him, and his father had died when he was very young. He was very attached to his mother, so he got into neuroscience strictly because he wanted to be able to replicate his mother's consciousness in some kind of a in silico device. So he figured we had to understand the brain if we wanted to do that. And that was the motivation for the first fMRI experiment, if you can believe it. But really, it's true. The first bold experiments were performed in a number of sites by Ogawa and the Minnesota group. We did it best because at the time we had the best scanner in the world to do echoplanar imaging. The other sites couldn't do echoplanar imaging. So their data was believable, but not great. So the first bold studies were done, Ken Kwong and Jack Beliveau using, these are the goggles that flash lights. And there you can see the activation of the visual cortex. But you can see that off, on, off, on. And what was paradoxical about the bold signal is Seiji Ogawa, in the first paper on bold, where he was doing hypoxia and stuff in animals, he predicted that you would see a negative bold signal in the visual cortex because you're going to be increasing deoxyhemoglobin. But it turns out because the blood is coming from the arterial side where you have very little deoxyhemoglobin, and oxyhemoglobin, as you may or may not know, is diamagnetic, so it has no magnetic signature, whereas deoxyhemoglobin is paramagnetic, so it has a magnetic signature. So in reality, what happens is when the blood flow comes in, it increases. Oh, well, okay, this is just give you an example of, you can also do the experiments with CBF too. But it increases the total blood flow, which brings in more oxyhemoglobin, which is diamagnetic. So therefore, you dilute the concentration of deoxyhemoglobin, thereby making the magnetic susceptibility match better between the vessels and the extravessel areas and the regular tissue, and therefore the signal goes up. So anyway, you can just see that you have very high contrast to noise here. So BOLD looks like it's going to be a good tool for mapping brain function. However, you can also, as I was talking about, use blood flow. So these are some studies that were done by Larry Wald and Rick Hogue. And what they did is they use a coil to do labeling of a single carotid artery. And you can see that you have the control and then the control minus the tag. And you can see you can pick up the territories of the brain that are perfused by that particular carotid artery. And then you can also do, say, in this case, a motor task. You can see that you can get the motor activation here in the cortex here, or you can do the visual task, and you can see that you get a much more constrained area in this map, t-value map, because BOLD, as we know, it includes a lot of the vessels and the veins. So the territory that you activate with a BOLD signal actually is larger than the actual area that's activated, and the blood flow is a better readout on the actual tissue activation. But as I told you before, because of the limitations of contrast and noise, most people still use BOLD. This is just an example some years ago that we did looking at switching between finger and toe movements. So what's nice is that you can really map out the somatotopy of the motor area using these types of studies. Here's another one, signing your name in the magnet. And this is just another fMRI study that we did in, looking at nicotine activation. And you can see that there are these two peaks associated with nicotine activation. And you can map out very nicely. And this is a map of functional connectivity, which I'll talk about a little later. You can map out the connectivity in the brain using fMRI. Now, this is using the iron technique, the iron oxide. This is a monkey. And we get data that's like way better than we would get using the BOLD technique in a single subject. And you can see, like, here's an interesting thing that shows that there's a nice correlation between the functional connectivity of the globus pallidus and white matter following a nicotine injection, which is kind of interesting. And then you can map out the regions of the brain that are connected together after following a nicotine stimulation. And then these are just the connectivities that you can see. A lot of times you'll see the functional connectivity as a circle with all these lines going across. I find those things so complicated and hard to read after you get past about 20 regions of the brain that it's just not worth it, but some people like it. So this has led to people hoping that what we'll be able to do is to get a connectome and of the brain using both the structural data from diffusion spectrum imaging or diffusion tensor imaging, and also from the connectome using functional connectivity. So this is a nice review article that was published a couple of years ago outlining the problems with doing structural and functional connectivity and putting them together. And so what you can see is you get regions of the brain. So this is a map of regions that are connected together. You often see these matrix type maps. And then you can see that you have anatomical projections and neural activity in these different brain regions. And so you're going to try and map together these two structural and functional connectivities to get an overall picture of what the heck is going on in the brain. And here is kind of the end result, which is that it turns out that the structural and the functional connectivities are very different. And this is for a number of different reasons. You have polysynaptic connections among neurons. And so the activity can spread outside of just what you would see as a structural network. The other problem we also have is that the structural connectivity that we find, we also can't map it yet at a high enough level of spatial resolution that we would like to be able to really map out all of the finer structural connections. And however, the technology is progressing in this domain. What you can see here is even for what we have now, the correlation coefficients that you get between the structural and the functional connectivity are not very high. As a matter of fact, 0.7 is really an upper limit in only a couple of structures. You're not going to see that across the brain overall. And so what you have to do is you have to be able to come up with different metrics for looking at functional connectivity or structural connectivity and models, statistical models, biophysical models, ways of trying to associate those two together to start to develop these combined maps. And this is certainly an area that's expanding in its purview and will be kind of one of the, I think, hot research topics in the next 10 years is putting these things together. And also bringing in other data, like MEG data, and even direct electrocortical recordings. So getting back to the, I realize now that this, probably should have had this before, but the oxyhemoglobin, this is just kind of a picture showing you what I was telling you before about BOLD, is that you've actually decreased the tissue concentration of the oxyhemoglobin, which is why BOLD goes up. And what's nice about if you combine measurements of BOLD and flow, you can actually start to measure oxygen consumption. And that is another interesting, parameter that we'd like to know about. And so there's ways to do this. One of the things you can do is you can increase flow using hypercapnia, which increases flow but doesn't increase CMRO2. And then you can repeat that in the same subjects, and then you can get the CMRO2 change. And this is kind of an example of that in the visual cortex from our lab, Rick Hogue, and a long time ago doing this. And you can see that you have the visual stimulation hypercapnia. They're pretty matched here. And the BOLD is unmatched. And that's largely because of the oxygen consumption that's happening. With the CDF, you're not getting that aspect of things. And so now you can start to measure these things like CMRO2. You can also model it using BOLD. And what you can see is, which is nice, is that as has been known in the literature from years before, but before MRI that is, that there's a tight correlation between the oxygen consumption and the perfusion increase during, say, tasks and activation of the brain. And so it's nice to know that, I think this coupling is very important, right? Because without this coupling, we wouldn't have as much confidence in being able to use fMRI as a readout on brain activity, because we know that the oxygen consumption really is a readout on where the brain activity is happening. There's a lot of problems in fMRI in terms of controlling variables, and these are kind of glossed over in the literature a lot. Blood pressure can when you go to extremes, that can affect the perfusion and the BOLD. There's also things like caffeine, nicotine, the menstrual cycle that can have huge effects on BOLD. For instance, caffeine and theophylline, which is another stimulant in T, it's a adenosine receptor antagonist. Those are vasoconstrictive. They can also affect dopamine release. And estrogen is also a potent modulator of blood flow. CO2 is very important. And then there's also psychological variables that can affect your baseline state, like craving, expectancy, and fear. And there's any number of studies showing. So these are things that you'd like to control when you're running your conventional fMRI experiments. This is an experiment done in our lab at a student we had, Greg Zaharchuk, where he was looking at what happens to CBF when you decrease blood pressure. And you can see it's fairly constant over a wide range of blood pressure, but once you start getting below 60, it starts to drop. This is an experiment we were doing in epileptics and controls, where we were looking at changes in hyperventilation, how that affected brain electrical activity. We were doing simultaneous EEG in the magnet with the fMRI. But what you can see, which is another thing that's important, is that so we had these people hyperventilate, and what you can see is that there's a direct correlation with the end-tidal CO2, and the signal drops when the end-tidal CO2 goes down. The signal drops. But what's interesting? Yeah. Yes.
No, that couple of minutes. If you can close with.
Yeah. Okay. All right. Sorry.
fMRI, you covered the fMRI, you covered the structural.
Okay, and then there's other things like nitric oxide, which are important in terms of determining the blood flow. There's other things.
For that, you need to come here. You need to come here, and we will do more hours of talks.
Sure. And nitric oxide is important, but also dopamine is not affected by nitric oxide. Dopamine is vasodilatory. And so this is an example of two dopamine molecules giving you positive and negative blood flow. But the idea is that one of the problems with mapping structural and functional connectivity is when you stimulate the brain, you can increase blood flow and you can decrease blood flow, increase BOLD and decrease BOLD. And I'm showing you here two examples of if you stimulate D1 receptors, you will increase blood flow and BOLD. If you agonize D2 receptors, dopamine D2 receptors, you'll decrease blood flow. And at any given point in time, these two things can be happening simultaneously. So you can come up with situations where you would see no change in blood flow, even though you had stimulation of receptors, simply because the stimulation of those particular receptors led to increases in blood flow or decreases in blood flow. And this is just another example of positive and negative blood flows. So I am going to stop there. And I just obviously it's a short talk and I only gave you a flavor. And I'm sorry it wasn't probably quite exactly as organized as it might have been.
No, that was great. That was great. I have a question for you.
Yes.
So for Risto and TMS EEG, what do you think is a way to efficiently or nicely involve fMRI connectivity, functional connectivity, and structural connectivity, DTI and functional MRI? Let's say if you stimulate, you have a hypothesis that you stimulate a particular circuitry. So how can you put together functional MRI connectivity and DTI, tractography, in your mind?
I mean,
with TMS and EEG.
Yeah. Well, I mean, you could do TMS, EEG, structural connectivity, and functional connectivity all in the same subject in the magnet. As a matter of fact, Tommy Rai in our lab, another Finnish guy, he is looking at TMS, Tommy Rai and Apo Numida. Apo. They're both doing TMS combined with fMRI in the magnet. And it's not inconceivable that you could wire them up with EEG electrodes, do TMS and fMRI and structural. I mean, it could be done. There's no physics reason why it couldn't be done. You would have interference with the TMS and the electrodes, EEG electrodes. So of course you'd have to blank out during those periods. But it's certainly possible to do all that. I mean, like I said, I've done a lot of EEG studies in the magnet in epileptic subjects. It actually worked way better than anybody ever thought it would. And I think that that this is just going to be a field that's, it's really kind of in its infancy right now, combining all these things. And I think that, to answer your question, it is possible to do it. And the question then becomes, how do you frame your questions? To be fair, doing TMS in the magnet is tricky and it requires a big investment of time and effort. Doing EEG in the magnet is by now, I think a lot of groups are doing that around the world. So as long as you're a kind of established research group, I don't think it would be a problem. But to combine TMS, EEG, and fMRI structural connectivity all in one scan, I mean, that's going to be, there's only a few groups in the world that can probably pull it off.
Yeah, but my question is, if you have a hypothesis for a specific circuitry, let's say we work with a stop signal task with inhibition, okay? Let's say in gambling disorder. This is a project that we work together.
Okay.
So you have a network that is the pre-SMA, the subthalamic nucleus connected with a hyperdirect pathway, and then you have the two gyri, two circumvolutions of the frontal area, frontal lobe, the inferior and the middle. Now you have these nodes. Russ Poldrack has written a lot about it, and Trevor Robbins. Okay, it's a lot of literature. It's 30 years of literature. more or less. Now, the question I have is, what do you gain? Because with all these excitations and inhibitions, this network is so complex with STN, with putamen, pallidum, thalamus. In the very end, what do you gain with all that? Can you really understand something more by adding fMRI and functional connectivity? Can you understand the sequence of events, causality or something with effective connectivity models? Or it's all, let's say, just increasing complexity and you cannot figure out anything?
Well,
No, I'm asking your sense.
I think, I mean, one of the things that you can do is you can manipulate things with either TMS and or drugs that can modulate expectancy, or can modulate craving, can modulate, you know, many aspects of function that would alter the functional connectivity in those regions. I mean, the thing about MRI, which is nice, is, you know, you're you know, mentioning all these brain areas, but most of these brain areas, these networks, have to be identified using fMRI, or you know, it's very difficult otherwise. And I mean, our lab, we've done a bunch of studies in drug abuse, for instance, looking at these complicated networks, and we've identified networks that really were very little described in the literature. For instance, we found these hippocampal, you know, limbic, you know, connections that you could pick up with the MRI, and then you can also see how they're modulated by, for instance, in our case, we were looking at abstinence, and you can see that these networks get modulated dynamically and with huge effect sizes just by changes in kind of, you know, baseline parameters of you know, elicited by abstinence. So I think
But this you're doing in rats.
that was in rats, yeah. But people have seen similar networks in humans too. Those same networks exist in humans. That type of limbic reward circuitry is very well conserved across species. But
You should have also some equivalence of behavioral tasks in rats. You know,
yes.
How are you discussing now frontal networks of the inferior frontal gyrus and middle frontal gyrus with a rat? I mean, it's very difficult. What I'm saying is probably it doesn't worth much of our effort even to get in these experiments with when you have TMS EEG and you stimulate, you get the recordings from the EEG. What do you want? And you have the structural connectivity. What would functional connectivity give you more?
Oh, well, all right. Okay. Look, I mean, that's a very open-ended question, right? So I can answer that question, though, in the case of the epileptic subjects that we've scanned, which is that it turns out that you can see a lot of the brain tissue. These things are hard to localize with the EEG. And by doing the functional MRI, you can actually start to pinpoint more accurately the regions where the actual epileptic foci are. And it's tricky in the magnet because, you know, it's hard to elicit a seizure in the magnet, but it turns out that these things leave metabolic signatures that you can kind of pick up. And so, you know, then with the structural connectivity, you could actually start to piece apart networks that maybe are affected by this particular epileptic focus. I mean, I know this is a very specialized application, but I also think it was, in this case, it was a very powerful one. And I think that there's, I mean, you know, I can't give you good examples of how TMS can be used in the magnet to really help you isolate a circuit using functional MRI. There's somebody in the room that probably could do that better than I. And there's people in our lab that could do it better than I. But I think that being able to manipulate, inhibit neural activity in the magnet while you're doing fMRI can only help you identify better which networks are affected by that particular you know, region. So I would argue that your question is saying, why is it necessary? And I could argue, why is it not necessary to be able to do this level of neural perturbation, you know, in the magnet, if you're interested in identifying, you know, a vast, a very complicated and detailed network.
Okay, Bruce, very good.
All right.
Thank you so much.
I know I didn't convince you, but, yeah, somebody else there will. Okay, thank you. Bye-bye.
Bye. So this kind of talk is more for you guys, because I asked him to discuss with him. That's very basic. It's not a function of what it is. Nothing more than that. But we didn't talk about